Welcome to Click for Biology. We're looking at plant nutrition and examining a number of experimental techniques. In this particular technique, we're going to be demonstrating the need for light. The experimental method that I will now describe is a classic experimental method and often comes up in examinations in a number of different contexts. If you would like a copy of this presentation, then you can find that linked as a PDF in the description below. To demonstrate the need for light, we are going to be using an indicator which is called hydrogen carbonate indicator, or sometimes it's called sodium hydrogen carbonate indicator, which is actually the same substance. Let's take a look first at how that indicator works. In the presence of low levels of carbon dioxide, the indicator turns a purple color. Low carbon dioxide levels would be indicative of a plant which is doing photosynthesis. If the indicator is in the presence of atmospheric levels of carbon dioxide, it is orange. This would tend to suggest that there is no photosynthesis. The alternative is that the hydrogen carbonate indicator turns yellow, and this would indicate that carbon dioxide is present in a high concentration, and therefore there is no photosynthesis, but there may well be something producing that carbon dioxide, which is likely to be respiration. Let's look at our experimental setup. We have a control. The boiling tube is sealed here, and the atmosphere of this boiling tube is now the same as atmospheric gases containing the atmospheric carbon dioxide levels. Notice that what happens in this situation is that the indicator has gone an orange color, and this suggests that there's been no net change in the carbon dioxide levels, and therefore there is no photosynthesis taking place. Not really surprising, given that there is no leaf present within the boiling tube. The second tube here, we're going to provide with light, which of course is the basis of our experimental technique. And within this tube, which contains atmospheric carbon dioxide, the leaf carries out the process of photosynthesis, using up the carbon dioxide, so the carbon dioxide levels are now low. As a consequence, the indicator turns purple. We then replicate this tube number 2, except that we're going to wrap around the outside of the tube black paper to exclude light. So now we're in the dark. The consequence of this is that the indicator turns yellow. The yellow indicates that there's carbon dioxide production. That carbon dioxide production is because of respiration. There is no photosynthesis because there's no light, and so carbon dioxide is not used up. This is a classic experiment, modified sometimes in different ways that we modify the boiling tubes to demonstrate different features. But here, the presence of light has demonstrated that photosynthesis is taking place, whereas in the dark, it does not. So 3 minutes of quick questions. Pause the video, write down your answers in the space provided. It should take you no more than 3 minutes. And then you can check out your learning and your answers by looking at the description where the answers are placed. And then we can move on to the next experimental technique video, which is linked here, the link to the channel to subscribe, and of course to the playlist. And if you have a moment, give us a thumbs up for the video, please.